Now, from a chemical standpoint, what we are looking at is carbon dioxide being recombined with water. Look at the equation above my head. Carbon dioxide, 6 CO2 plus 6 water, gets made into a sugar, C6H12O6, and 6 oxygen. Okay, notice that this is not being powered by ATP. No, this is being powered by sunlight. Now, when it comes to the reactions of photosynthesis that, make this equation happen, there are two pathways that we're going to focus on. The first ones, the ones that we refer to as the light reactions. The light reactions these are the reactions that are going to be powered by photons of light, hence the name, light reactions. When you look at the light reactions, they are not responsible for the making of sugars. What they are responsible for is the production of ATP. So light reactions harness solar energy. They will utilize that solar energy, as we're going to see over the next couple of lectures, to excite electrons. Excited electrons are then going to pump hydrogens, and those hydrogens are going to flow through an ATP synthase. All that ATP is then going to be used for this right here, the Calvin cycle. The Calvin cycle is where a series of reactions that are going to take carbon dioxides along with some pre-existing molecules, recombine, and ultimately end in sugar. To power this recombining and the splitting off and everything, the cells are going to need a lot of ATP, which comes from number one, the light cycle. Everything we're going to be talking about here is going to be happening in the chloroplasts. Okay? The light reactions are going to be occurring in and on the thylakoid membranes. Remember, the thylakoids are the green pancake-like things that are found in chloroplasts. This is where the reactions are going to be occurring. This is where chlorophyll is. Going to capture the energy from photon, transfer it to an excited electron, all of that. The ATP, once it's made, is going to then be utilized here in the stroma. Remember, the stroma is the aqueous solution that fills in the chloroplast, not the cell, the chloroplast. So when we look at the chloroplast structure, just as a refresher, we have an inner and outer membrane, okay, dual membranes, just like the mitochondria, except here, instead of a matrix, we have a stroma, the liquid-filled, the aqueous solution-filled inner volume. And then we're going to have stacks of thylakoid discs. That's what the green pancakes look like. Now, the thylakoids are hollow. So you're also going to have a separate area within them. So this way we can pump things back and forth between the stroma, across the thylakoid membrane, into the thylakoid lumen. We can set up differences.